Welcome to this explainer. Today, we're going to decode the high-yield rules and, honestly, more importantly, the critical exam trap exceptions of medical parasitology. Look, if you've ever felt completely overwhelmed by the sheer number of parasitic worms, their bizarre life cycles, and those endless lists of clinical presentations, you are in exactly the right place. We're going to bring some serious order to the chaos.
It's kind of a massive biological mystery, right? Like, why do some of these worms trigger huge immune alarms in the human body, while others manage to hide perfectly in plain sight, literally living inside us for years without making a single sound? Well, to answer that, we really need to understand exactly how these parasites operate, how they reproduce, and the incredible evolutionary strategies they use just to survive inside a human host.
All right, let's just dive right in by defining our main suspects here, the helminths. The term helminth is really just a catch-all for parasitic worms. In medical parasitology, we mostly divide these invaders into two giant phyla. First up, we've got the nemathelminthes, which are your classic roundworms. And second, we have the platyhelminthes, which are the flatworms. Grasping this basic division is absolutely your first step to mastering the clinical stuff.
So here is our roadmap for today's explainer. We'll start with meet the helminths, then anatomy and reproduction roles, the trematode life cycle, demystifying eosinophilia, and finally, ultimate exam traps. We're going to focus heavily on a rule breakers framework today. Because, in biology, knowing the standard rule is really only half the battle. Effortlessly spotting those tricky exceptions is what's actually going to secure your understanding. Let's get into it.
Section 1. Meet the helminths. Nematodes, cestodes, and trematodes. Let's start by looking at their foundational structures. You can essentially bucket the structural differences of these worms into three main groups. First, you have the nematodes. These are your non-segmented, perfectly cylindrical roundworms. Then, you've got the cestodes, or tapeworms, which are flat but heavily segmented. And finally, we have the trematodes, the flukes, which are uniquely leaf-shaped flatworms. Just visualizing their physical shape gives you some immediate, super helpful clues about how they actually interact with the human gut.
Now this next bit is where it gets really interesting, and honestly, it's where exam writers just love to set traps. Look at how their guts evolved. Nematodes have a completely normal, full intestinal tract, mouth at one end, anal pore at the other. Trematodes, on the other hand, have what we call a blind intestinal tract. They have a mouth, sure, but absolutely no anal pore. So whatever goes in must come back out exactly the same way. Yikes. And cestodes? They don't even have a functional gut at all. They just absorb nutrients directly through their skin, or tegument.
And speaking of cestodes, you should really think of them as the alternate gutless freeloaders. Since they don't have a digestive system to process their own food, they just hang out in our intestines and steal what we've already done the hard work of digesting. Talk about a free lunch, right? To pull this off, they use this specialized head called the scolex, which is equipped with hooks and suckers to hold on super tight to the intestinal wall. The rest of their body, it's just this long chain of proglottids. These are individual hermaphroditic segments dedicated entirely to pumping out eggs, which then break off and are passed in human feces. Moving right along to Section 2, Anatomy and Reproduction Rules, highlighting the exceptions. This is where the biological rules are well-defined, but spectacularly broken. So here's your foundational biological baseline. The general rule of thumb is that roundworms, the nematodes, are sexually dimorphic. That just means you've got separate male worms and female worms, and they actually need to find each other to mate. But the flatworms, both the cestodes and trematodes, are generally hermaphroditic. They carry both male and female sex organs, so they can essentially just do it all themselves. But watch out for this one. It is a massive exception, and honestly, it's one of the most highly tested concepts in all of parasitology. Schistosomes, which are blood flukes, are trematodes. So based on the rule we just learned, they should be hermaphroditic, but they aren't. They are the only trematodes with sexual dimorphism. They literally pair up for life. The slender female worm physically lives inside a little groove on the male's body called the gynecophoral canal. It completely shatters the flatworm role. And flipping things over to the nematode side, we find another really fascinating rule breaker. Remember, roundworms are supposed to have separate sexes, but Strongyloides stercoralis breaks the mold. They have hermaphroditic adult females that live in the human intestine and don't even need a male. They reproduce via parthenogenesis. So if you ever see a hermaphroditic roundworm mentioned, boom, you instantly know it's Strongyloides. Okay, here is a really crucial concept, the multiplication rule.
As a general rule, helminths simply cannot multiply inside humans. If you swallow one egg, you get one adult worm. That's it. To get a heavier infection, you'd literally have to ingest more eggs or larvae from the outside environment. But again, look at our exceptions. Strongyloides storcoralis and Capillaria philippinensis are the rare, terrifying rule breakers that can auto-infect humans. Strongyloides is especially notorious for causing deadly hyperinfections in immunocompromised patients, precisely because it's able to reproduce and multiply its numbers entirely within the host. Section 3. The trematode life cycle, going from water to host. Let's see how they make this highly testable journey. Let's track this incredible journey of the flukes. Step 1. Eggs are passed out of the human host in feces, urine, or sometimes even sputum. Once they hit water, these eggs hatch into a free-swimming stage called miracidia. Now, these miracidia have one singular mission in life: find a very specific species of aquatic snail. Snails are the obligatory intermediate hosts. It's where the parasites actually multiply. Finally, the snail releases a brand new stage called cercocaria, back into the water, armed and completely ready to infect a human.
And depending on the fluke species, those cercaria use incredibly diverse methods to breach our defenses. Schistosomes, they're the direct assault team. Their free-swimming cercaria have these little forked tails, and they just directly penetrate bare human skin in contaminated water. But liver flukes, like Clonorchis or Fasciola, are way stealthier. They turn into cysts called metacercaria and hide on aquatic plants or inside freshwater fish, just waiting for us to eat them. Lung flukes like Paragonomus do something similar, hiding out inside crabs or crayfish. So the route of infection tells you exactly which parasite you're dealing with.
Section 4. Demystifying eosinophilia. This is the body's tissue alarm system. Let's look at how the body responds to these invasive parasites.
Let's define the term real quick. Eosinophilia is basically a massive surge in a specific type of white blood cell called an eosinophil. In the context of parasitology, you can think of it as a cellular siren. This alarm goes off specifically when parasitic larvae physically breach and invade human tissues. It is the hallmark biological sign of invasive helminthic infections. If you've got a patient with an unexplained high eosinophil count and a recent travel history, helminths absolutely need to be at the very top of your differential diagnosis.
But here's the catch. You have to know when that alarm actually sounds. The severity of eosinophilia correlates strictly with the degree of tissue invasion. The alarm is at its absolute maximum during larval migration, like when worms are actively tearing their way through tissues like the lungs, causing conditions like Loeffler syndrome or Katayama fever. However, if adult worms are just hanging out safely in the empty space of the intestinal lumen like an adult tapeworm, the alarm is essentially silent. Similarly, if the worm is perfectly walled off in a tissue cyst, like an echinococcal cyst, no antigens are exposed to the immune system at all, and eosinophilia is completely absent.
Finally, Section 5, Ultimate Exam Traps, featuring the rule-breaking protozoa. Let's bring it all together with a final set of exceptions guaranteed to test your precision.
So, we've spent a lot of time on worms today, but what about the single-celled parasites, the protozoa? Well, as a strict biological baseline, protozoan infections generally bypass that whole eosinophil alarm system entirely. If a patient has a protozoan infection, something like malaria, giardia, or amoebiasis, you generally do not expect to see eosinophilia. But as we've learned, biology loves throwing a curveball, so make absolutely sure you memorize these 3 specific protozoa that brilliantly break the rule and actually do trigger eosinophilia. Number 1. Cystocyspora belli, an intestinal protozoa that causes eosinophilia in immunocompetent hosts. Number 2, Dientamoeba fragilis. And number 3, tissue-borne Sarcocystis species. If you ever see a case with a protozoan and eosinophilia together, it is almost certainly going to be one of these 3 suspects. Which really brings us to a vital philosophy for understanding all of this material. The standard features, they're great for building a foundation, but to truly grasp clinical parasitology, you have to look past the normal operations. You have to actively hunt down these specific anomalies. It's the rule breakers that actually define the boundaries of medicine.
So I'm going to leave you with this to ponder. For a parasite, the ultimate goal is survival, right? Which usually means staying entirely under the radar. So what evolutionary advantage do migrating larvae actually gain by being so violently destructive that they trigger our loudest, most aggressive immune alarms?
It's a wildly fascinating evolutionary puzzle about that delicate, sometimes violent dance between host and invader. Thank you so much for joining this explainer. Keep questioning the rules, and we'll catch you next time.